Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Neisseria, gonorrhea. Here to remember the spelling, spell like this: gonorrhoeae, rho, R-R-H-O, rho, E, single E, A for aeroplane A, E. This organism name is complicated spelling: Neisseria gonorrhoeae. It is an obligate human pathogen. It means it lives only in the humans. So, source of infection may be other infected human or asymptomatic female carriers mostly. It is the causative organism of venereal disease gonorrhea. This spelling is very simple: gonorrhea, single A. It is the second most prevalent sexually transmitted bacterial disease. Then what is the first most prevalent sexually transmitted bacteria? It is chlamydia. It causes non-gonococcal urethritis. Here, gonococcal urethritis is second most prevalent venereal disease, nothing but sexually transmitted disease. Coming to its biology, it is a gram-negative cocci, hence it is also known as gonococcus. Fastidious, which means it requires special media and special requirements, like it only grows on enriched medium, it won't grow on normal agar. Enriched medium like chocolate agar. And it requires carbon dioxide. It is capnophilic. I already told you it lives only in humans. Why? It can't survive in environment extremes of temperature. So it requires a temperature of 37 degrees, 35 to 37 degrees, and carbon dioxide of 3 to 7%. Remember like this: 37 degrees, 3 to 7% carbon dioxide, 6.5 to 7.5 pH. With all these requirements only it will grow, hence it is called fastidious organism. It is non-capsulated but have a cell wall. It is non-flagellated but have a pili. It is non-sporulating but it produces endotoxin. It is arranged in pairs. A pair of cocci is called diplococci with concave. That means here you can see concave adjacent sides appears in a characteristic kidney or coffee bean morphology within polymorphonuclear leukocytes, that is neutrophils. Different strains show heterogeneity, means variations genetically and phenotypically. Why? Because of extensive genetic exchange intra-strains within the strains and inter-strains within species also. It will show extensive genetic exchange, hence it will have high variations. This high variability is valuable for evasion and development and spread of antibiotic resistance. What is meant by evasion? Evasion, E for evasion, E for it escapes recognition by immune system. Anyhow it escapes from our immune system. That is nothing but evasion. Why? It has high variability both genetically, phenotypically, because it involves in extensive genetic exchange, either by means of transformation or conjugation, whatever it is. If antigens are varying, our immune cells can't recognize them. Next, coming to structure, coming to genetic material through which it is transmitting this property, antibiotic resistance property, from one strain to different strain and developing the antibiotic resistance through plasmids. Which plasmid? Beta-lactamase or penicillinase encoding plasmids. We will give penicillin to destroy Neisseria gonorrhea, but it has a plasmid with it, which is nothing but penicillinase or beta-lactamase. It binds to cyclic amide bond of beta-lactam ring of penicillin and destroys penicillin. So penicillin can't do anything. Hence it is called penicillin-resistant Neisseria gonorrhea because of the presence of this plasmid. If this plasmid is transferred to other strains also, those strains which are taking this plasmid also will get penicillin resistance. That's why its genetic exchange is important to transfer antibiotic resistance to different strains of same Neisseria gonorrhea. These conjugative plasmids transfers this penicillinase plasmid to other Neisseria gonorrhea strains. Ultimately, every strain will get its property through extensive genetic exchange. Cryptic plasmids, which means its function is hidden or not known. Three types of plasmids are there. One is beta-lactamase encoding plasmid, providing resistance against penicillins. Conjugative plasmid, which helps in transfer of this plasmid. Then cryptic plasmid, we don't know what function it is there. It is conserved. Next, coming to cell wall. I already told you it doesn't have capsule, but it possesses a cell wall, which consists a gram-negative bilayered outer membrane, which has many proteins like pilus, porin protein, PorB, Lipooligosaccharide, LOS, opacity proteins and other proteins. We will see one by one all these proteins present in outer membrane, which is gram-negative bilayered membrane of cell wall. These structures have been extensively studied to elucidate pathogenesis, virulence, candidates for diagnostics or vaccine production, phenotypic and genotypic characterization. What is meant by pathogenesis and virulence? If a bacteria has a capacity to infect, it is called a pathogenic bacteria. If it is not infective, it is non-pathogenic bacteria like commensals in our mouth, gut, which produces vitamins and helps us. Friendly bacteria, which are non-pathogenic, they don't possess a quality of infection. So it is qualitative, whether it is pathogenic or not. Virulence is quantitative. A pathogenic bacteria, how, to what degree, to what extent, to what severity it will cause a disease, it is called virulence. Example, PorB proteins are of two types, B1A and 1B. 1A involved in local infection only, whereas 1B, it can cause extensive disease and dissemination, so highly severe. So it has more virulence. Virulence is related to quantity, how much severely it causes a disease. Pathogenicity means it will cause or it won't cause a disease. That's it, qualitative. Then using these proteins, we can develop a diagnostic test, or we can develop vaccine or not to elucidate. Using these proteins to differentiate, to characterize different strains phenotypically and genotypically. Coming to peptidoglycan layer. Outer membrane contains various proteins. Finished. Below that outer membrane, there is a thin layer. All gram-negative bacteria will have a thin layer of peptidoglycan, whereas gram-positive bacteria will have thick layer of peptidoglycan in the periplasm. Periplasm means space between outer membrane and inner membrane. So here peptidoglycan layer lying below outer membrane and above inner membrane. In between these two membranes, the space is nothing but periplasm, which contains thin layer of peptidoglycan. What peptidoglycan contains? N-acetyl glucosamine, N-acetyl muramic acid connected by peptide bonds, glutamic acid, diaminopimelic acid, alanine. Then coming to inner membrane, nothing but cytoplasmic membrane which envelops cytoplasm. What cytoplasm contains? Solutes dispersed in viscous solution. What bilayered membrane contains? Penicillin binding protein 1, penicillin binding protein 2. In the name itself it's there. If we give penicillin, it will bind to this. What is the function of these two proteins? Its function is to produce the layer above it. What is the layer present above it? Peptidoglycan layer. What it contains? N-acetyl glucosamine, N-acetyl muramic acid connected by peptide bond. So it produces and maintains peptidoglycan layer. Now penicillin is binding to these proteins. What penicillin will do? It destroys peptidoglycan production. It won't allow it to produce and it will destroy it. Hence penicillin is attacking on peptidoglycan layer of our Neisseria gonorrhea by binding to penicillin binding proteins in cell membrane. So against the penicillin, these have plasmids. What plasmids? Penicillinase encoding plasmids, which destroys beta-lactam ring of penicillin. Now we will see outer membrane proteins one by one and finish. First, Pili. We all know it is a hair-like appendage made of thousands of pilin, which is nothing but protein subunits. Pili extends several micrometers outside the cell. It is a principal virulence factor. First protein we are studying, Pili. It is principal virulence factor associated with adhesion of epithelial cell. To infect a cell, it has to first attach to it and then invade it, enter it. So, first attachment, adhesion. Then it prevents neutrophilic phagocytosis. It has to evade our immune system. That means escape our immune system through preventing neutrophilic phagocytosis. Then transformation of exogenous DNA. Pili forms conjugative tube in between two bacteria and helps in transformation. Here, this Pili takes up the exogenous DNA. Dead bacilli will release DNA and will die. Those DNA which have any antibiotic resistant properties or some other properties useful, then it will take it into it. This is nothing but transformation of exogenous DNA. It is done by Pili. It has three properties. First, adhesion, then evasion by preventing neutrophilic phagocytosis, then transformation of exogenous DNA into the cell from outside. Next protein, porin protein. In the name itself it's there, it forms pores. It is nothing but PorB protein. It is universally present in bilayered outer membrane. That means it contributes to more than 50% of outer membrane proteins located in close proximity to lipooligosaccharide and reduction modifiable protein. What is the structure of this porin forming protein? Pore forming protein, trimeric pore forming transmembrane protein. It means it crosses the membrane, transmembrane, pore forming. It forms pore in the outer membrane. Trimeric structure. It has three structures attached forming a pore that allows solutes ions pass through outer membrane. Common sense. PorB is target for antibodies, which means this porin forming protein is immunogenic. It is inducing the host to produce antibodies against it. Involved in same, there also first protein adhesion, second protein also adhesion. Here extra thing is invasion. Evasion of complement mediated destruction. It also inhibits neutrophils. Both first protein, what is it? Pili. Second protein, porin. Both are anti-neutrophilic. Inhibits neutrophils by interfering with its activation, degranulation, phagocytosis. And then it increases its survival only inside the cell. It enters neutrophil. Neutrophil tries to kill it, but it interferes with its degranulation, phagolysosome formation. Then it improves its survival inside the cell. Those cocci only we can appreciate as pairs, kidney shaped diplococci, gram negative. Then this porin induces apoptosis. It can translocate into cell membranes of host target cells, that is immune cells like neutrophils, macrophages, and disrupts its cell mitochondrial membrane and leads to its death, apoptosis. From bacteria, it translocates to our host target cells, that is immune cells, macrophages, neutrophils, or epithelial cells, and disrupts its mitochondrial membrane, leading to its death, nothing but apoptosis. Here, extra function of pore forming, it forms pores in our host cells also and causes its death, apoptosis. Adhesion, invasion, apoptosis, and inhibits neutrophils. I already told you while telling about virulence, PorB has two types, 1A, 1B. 1B is more virulent. That's why it is causing disseminated disease. 1A is less virulent. So, degree of severity is less. So, it causes only local infection. Each strain contains either 1A or 1B. No strain will contain both at a time. Then, Lipooligosaccharide consists of in name itself, it's a lipid moiety, which confines the endotoxic activity. I already told you it produces endotoxins. So here lipid moiety produces endotoxin activity, eliciting host immune response. That is, it is immunogenic. Core oligosaccharide, which contains glucose, glucosamine, galactose, galactosamine, and KDO. Lipoooligosaccharides are targets for antibodies. That means it is also immunogenic, like porin, involved in like porin, adhesion, invasion, toxicity of epithelial cells. And it escapes our antibodies. How? By sialylation and increasing antigenic variation. If one antigen is varying into another antigen showing variations, our immune cell can't detect it to test try. And by sialylation, what is meant by sialylation? Adding up a sialic acid to itself, like our human cells. Host cells will also have sialic acid. So, antibodies will detect it as a part of our own body cell and it won't destroy it. That's why evasion is possible by sialylation of lipooligosaccharide. Opacity proteins, name itself it's there, it provides opacity to colonies in specific culture medium. Involved in same adhesion, invasion of epithelial cells, neutrophils. How here it will evade opacity proteins? Opacity producing genes have 12 types of genetic loci. So it produces 12 antigenetically different proteins. At a time 3 to 4 different antigenically proteins of opacity proteins will be there. So it will escape our immune system through this mechanism by antigenic variation. Coming to other proteins like RMP, reduction modifiable protein, evasion by immune interference. Here it has an interesting characteristic, immune interference. By attracting non-bactericidal antibodies, masking it. Here, our reduction modifiable protein induces production of non-bactericidal antibodies. Those antibodies won't cause anything to the bacteria and will come and mask the bacteria. So, bactericidal antibodies can't detect this bacteria, hence it is safe. So, producing evasion means escaping from bactericidal antibodies using non-bactericidal antibodies, reduction modifiable protein. It is a conserved and immunogenic protein. What type of immunogenicity? Not true bacteria, not true bactericidal antibodies, non-bactericidal safe antibodies it will induce to produce and will hide in between those non-bactericidal antibodies and bacteria will be safe. Then other protein is IgA1 protease. Name itself it's there, it cleaves IgA1 antibody which is mucosal antibody. Hence it survives in mucosa infecting human hosts. Now only thing we have to learn is In outer membrane protein, various proteins are there. Each of the protein have its own properties. Pili, porin, lipooligosaccharide, opacity protein, and other proteins like reduction modifiable protein, IgA1 protease. In everything we can write attachment, invasion, evasion, and neutrophilic inhibition. Pili has extra property, transformation of exogenous DNA. Porin have extra property, it forms pores and allows solutes and ions to pass through the membrane. And porins have extra property of apoptosis. These pores, it will form in host cell also and leads to its death. Then lipooligosaccharide, extra property, endotoxic activity and sialylation. Opacity proteins, high variations and it will survive. Reduction modifiable protein induces non-bactericidal antibodies. It won't cause anything to bacteria. Non-bactericidal antibody. We'll revise entire thing with this diagram. I told you hair-like structure made up of thousands of pilous subunits, nothing but pili. Then trimeric. Here which structure is having 3 structures attached to it forming a pore? This green colored one. Nothing but pore forming protein PorB. PorB is in close proximity to reduction modifiable protein and lipooligosaccharide. Here the orange color is reduction modifiable protein. Here this one thread-like structure is lipooligosaccharide. Then what is left out protein? We studied pilus. We have seen green color porin. We have seen orange color orange green color porin, this one thread-like structure lipooligosaccharide, orange color reduction modifiable protein. Only protein left out is opacity protein. This blue colored one is opacity protein. All these proteins are present in outer membrane, which is present abundantly, contributing more than 50% of proteins. Porins. Hence, it has 3 structures attached to one another and forming a pore. It is major or principal outer membrane protein. Hence, it is given old name major outer membrane protein or principal outer membrane protein. Then coming to thin peptidoglycan layer, it has NAG, N-acetyl glucosamine, peptide bond linking with N-acetyl muramic acid. N-acetyl glucosamine, N-acetyl muramic acid peptide bond. Here it is thin because it is gram-negative bacteria. Then cell membrane. Here I have drawn in pink color only peptidoglycan and pink color only these 2 proteins in inner cell membrane. What is this? It is nothing but peptidoglycan producing penicillin binding protein 1 and penicillin binding protein 2, which produces and maintains peptidoglycan layer, which is the receptor or attachment site for our antibiotic penicillin. Remember my channel name, The Average Dermadoc. Don't forget to subscribe and share. Thank you. Hasta la vista. Dasvidaniya.